Okay, so we are transitioning out of basic characteristics of microbes, and now we're going to start talking about microbial growth. So over the next 3 weeks, our goal is to cover just general characteristics of growth and how do we define microbial growth. And then we'll move into talking about how do we control growth using different physical and different environmental and different chemical pressures. This will be a conversation that doesn't include antibiotics. We'll save that for a little bit later. And then we're going to spend a class period or two talking about microbial communities, in particular talking about it in the context of the human microbiome and how we can look at the human body as basically a variety of different microenvironments where there is selection for a certain type of bacteria based on these growth characteristics that they have. Okay, so today's lecture is pretty terminology heavy. And these are terms that we're going to be using over the next couple of weeks. So I just wanted to give that fair heads up. And the homework assignment kind of illustrated that. And having each person define a set of 4 or 5 terms, those are the terms that we're going to be using to describe these microbes. And actually, in lab, you may be able to identify your unknown by the end of tomorrow. The next step in the process is that we are going to go ahead and measure the growth of your unknown over a range of different salt concentrations, range of different temperatures, pH, and we're also going to test for oxygen requirements. And so you'll be able to define your unknown as being, for example, a non-halophile or halotolerant organism based on the characteristics that you observe for it. So where I thought we would begin today is just with a really simple example of a typical bacterial growth curve. And one of the things that we'll talk about on Wednesday is how do we actually measure growth? And that'll be kind of a prelude to the next couple of days in lab afterward. But a typical bacterial growth curve, there we go. Oh, damn it. All right. So we'll put CFU per mil on this end. And what this is it's a measurement of colony-forming units per milliliter. The CFU, or a colony-forming unit, we assume each colony on an agar plate has arisen from one bacteria. And you'll get a look at this in the next couple of days. But right now, just think of this as a measurement of bacterial concentration. And on the x-axis, we'll put time. Normally, when we're performing growth curves in a laboratory setting, we're doing this inside of a test tube like the ones that you've been working with. And what do you see? When you look at that test tube normally, what's the observation? Has anybody just gazed into one of those test tubes and wondered, what the hell is going on? Probably not. What do you observe? Think back to any of the cultures you've worked with. What's it look like? It's cloudy, all right? And that represents bacterial growth. So one of the ways that we can measure this is by measuring how much light is passing through these cultures. And that's something we'll talk about a little bit later. But usually when we're looking at these bacterial cultures, we're looking at about 24 hours out. And it's not as if it just goes to that. What do you have to do to start a culture? All of you have done this in the past week or so. Inoculate it. So you start from a single colony, you inoculate that into broth, and somewhere in between that starting and their end, there is this period of rapid growth, and then it peaks out. And usually what we see is just what happens at the beginning and the end. We're not watching what happens over those 24 hours in between. Now, each of these different phases of growth have a name associated with it. When we typically see our cultures out here, what is this stage known as, or this phase of growth known as? Yeah. This is stationary. So stationary phase, sometimes just referred to as S, is defined as a period of time in bacterial growth where the amount of nutrients are limited. Bacteria have pretty much eaten all the nutrients that are available within the test tube that we provide for them. The amount of new cells forming is equal to the number of cells that are dying. And so that's why there's no change in concentration over that period of time. If we were to follow out these cultures for another 3 or 4 days, we might see the addition of another stage known as death phase or death stage, where the amount of death is greater than the amount of cells that are replicating. At this point, usually we have toxic byproducts accumulating as well. And actually, we've talked about a couple of these toxic byproducts, but not defined them necessarily as such. Fermentation. What are the end results of fermentation? Or potential? What was that, Emma? Lactic acid, so low pH condition. What else? What if it's alcoholic fermentation? Ethanol and CO2. Ethanol is another toxic byproduct to microorganisms. And so the accumulation of those additional byproducts can lead to this observed death phase as well. Okay? But what's happening before then? What happens within the first 24 hours? What's this rapid growth period known as? Exponential, this depends on which side of the Atlantic you're on. So if you're in the UK, they will insist on exponential. If you are in the US, we will also take the word log. So it just depends on who you're talking to. Defined as rapid growth, this is the cycle at which at a given temperature, the amount of bacteria that are forming is at its maximal reproduction rate. For some bacteria like E. coli, this can be as short as 20 minutes. 20 minutes to go from 1 cell to 2 cells, or from 2 to 4. And that for E. coli is at about 37 degrees Celsius or so. The process by which this occurs, what is the process of division in bacterial cells known as? Starts with a B, ends with a fission. Binary fission, okay? So 1 cell becomes 2. And basically the process of this, cell increases its length or size, so we have to have additional peptidoglycan forming, additional cell membrane forming. We have duplication of the genomic material, and those chromosomes in plasmids will go to opposite poles of the cell, allowing for that binary fission to occur. Last phase, which is actually the first in this process, known as what? Yeah, this is our lag phase. So what's the definition of lag phase, or what's happening during this period of time? In some ways, yeah, right? No bacterial growth. But there's metabolic activity still within the cells at this point. Oftentimes what we find in lag phase is that cells are adapting to their environment. If we were to look at this at a molecular level, what's happening at the DNA is this is a point at which cells are beginning to perform transcription and translation of proteins that are required for growth in a given environment. So for example, let's say you have an organism growing in the presence of oxygen. What form of metabolism is most likely to be used when oxygen is present? Aerobic respiration, right? Now let's say you move the microorganism to an environment without oxygen. What could happen? Anaerobic respiration, and what else? Fermentation. Does fermentation use the same set of genes or go through the same pathways as aerobic respiration? Glycolysis is shared, and that's about it, right? So in order for a cell to adapt to that changing environment, it has to change the genes that's going to be expressed to allow for different pathways to be activated. That's exactly what's happening at lag phase. And so we can observe a cell, if we were to continue to change the environment again and again, we could actually observe new lag phases with each of those changes that we apply to it. Okay. So this is a typical growth curve. One of the things we're going to begin to look at in the next few days is how does this growth curve change when we start to change environmental conditions. And we're not going to track every hour what happens to the growth. We're going to do an endpoint analysis. So let's say, for example, this represents the growth of E. coli at 37 degrees Celsius. E. coli is a mesophile. Okay. Here's what I want you to do. Based off of this curve here, draw for me what you think the growth curve would look like. And typically when we do measure, well, I'll come back to that. Show me what you think the growth curve would look like at 22 degrees Celsius and 60 degrees Celsius. I'm going to say one more thing here, and that is typically when you're looking at the ability of an organism to grow over a range of temperatures, we're really oftentimes only analyzing what happens right here. Okay, so for example, the book shows mesophiles grow over a certain range of temperatures. Their y-axis is growth, the x-axis isn't time, it's the particular temperature. All right, so what we're going to try to do in the next few minutes is how do we relate this graph to the ones that we would see in the book. But draw for me what you think a growth curve would look like for E. coli under both of these temperature conditions. What temperature do mesophiles grow best at? So for those of you that defined this one, what temperature do mesophiles grow best at? Neither Fahrenheit or Celsius. I'll take either. I know you answered this one. What's body temperature? Yeah, 98.6 is one estimate, 37 is Celsius. I'm going to use Celsius because that's what I'm used to using when it comes to scientific stuff. But 37 is optimal for a mesophile. Okay? What's its range? 20 to 40, roughly. So what would happen at 22? Would the organism be able to grow? Yes, but how would that growth curve look? Slower? Absolutely. So at 22 we might find a couple of things. Might actually find maybe a longer lag phase. That rate of growth might be slower. Might not reach the same optical density at the end of it. Okay. How about 60? Is this within the range that supports growth or outside the range? So what would we expect to see then, Emma? Yeah, flat line, right? So at a higher temperature like 60, this, or what might be the alternative? Yeah, death, decrease. So we might actually see something more like goes down to 0. Now, when we talk about these growth ranges, when it comes to temperature, salt concentrations, and why am I blanking on the last one? pH. When we talk about those ranges, these are ranges that support growth. Okay? We're not talking about ranges that begin to kill the microorganism. And those are slightly different questions. So you might find, for example, that an organism like a mesophile can grow best between 20 and 40 degrees Celsius, 45 somewhere in there. Optimal is at about 37. Could they survive at colder temperatures than that? Yes. They could potentially survive at colder temperatures than that, but it doesn't mean that they're going to grow. So there's a slight difference here when we're referring to growth versus survival. Today, we're just focused on growth. Wednesday, we're just focused on growth. Eventually, we're going to start to talk about growth inhibition and how to actually kill them. And that's where we want to go to more of the extremes. So let's take a look at this then in a different way. I want everybody to try to do a manipulation real quick, mind-wise. And now what we're going to do is we're going to basically take these data points here, and I want you to plot for me just theoretical data. If we have our y-axis as CFU per mil, so concentration of bacteria, and our x-axis now is temperature, how does this curve look for a mesophile? Okay, so how does that relate to this data right here? And so really all we would have to do, so concentration over here, temp, and if we just plotted those three, for example, 22, 37, 60, we said there was no growth at 60, or really maybe those cells would die. Maximal at 22, or sorry, 37, and 22 there might be some growth. Okay, and if we were to connect these, then it looks something like that, roughly. Okay. This is typically what we see in the book. And this is what you guys will actually be graphing when you look at your particular unknowns. So a plot like this then allows us to determine whether an organism is a mesophile, thermophile, hyperthermophile, psychotroph, or psychrophile based on the range and also its optimal growth temperature. Now sometimes things don't fit really nicely into all of these boxes. Sometimes you might have one that falls somewhere in between, and you have to make a judgment decision in those cases, and I understand that those exist. What I'm looking for are strong arguments that you're using the data to support your decisions, okay, that they make logical sense. I'm going to pause for a minute then and just ask, what questions do you guys have about the terminology? And there are a couple that I'll hit on, but questions about the terminology that we're using. Go ahead, Emma.
When building up psychotrophs and psychophiles,
Yeah.
do we need to know the term, it's like extremophilic or something?
No.
Okay.
No. So.
Like it would like say it was a type of that kind.
Yeah. So the my guess is that extremophile is probably the term. So extremophile is kind of a broad term referring to those that live outside normal ranges, quote unquote normal ranges. So like organisms that live at a pH of 1, or organisms that live at a temperature of 105 degrees Celsius. Those are what we would call extremophiles. Oftentimes what we find are those extremophiles are not bacteria, they're actually archaea. And so that's one side note. The other thing that we should think about real quick too is which ones, for health reasons, what are the characteristics of organisms that we should be most concerned with? What temperatures? What salt concentrations? What oxygen ranges? What pH values are we most concerned with? We're concerned with those that are going to infect the human body. So what's body temperature? 37-ish, right? We're concerned with mesophiles because if we were if a hyperthermophile made it into our body, can a hyperthermophile grow at 37 degrees Celsius? Nope. No growth equals no symptoms. Okay, typically. What about pH values? What pH values might we be concerned with for organisms that we consider to be pathogens? Are we concerned with neutrophils, acidophiles, achillophiles? Both, actually. Both. Neutrophils and acidophiles. They have to be. Why?
Acidophiles, some of them are found in your stomach, but
Yeah. And that's the key barrier, right? The stomach is a barrier that exists to, in part, digest food, but also it prevents infection. So we've got some organisms like Helicobacter pylori, which is an organism that's acidotolerant. It secretes sodium bicarbonate as a way to prevent it from being destroyed by the low pH of the environment of the stomach. Others, they have to be able to at least survive that condition for about 8 hours, which is the amount of time it takes for food to be digested and go through the gut. And then once they get into the small intestine, pH levels start to rise. What else happens what happens to other environmental cues as we go from stomach to small intestine to large intestine? What else might change in those? And think about the 4 major conditions that we're focused on: temperature, pH, oxygen, and salt concentrations.
can you ask again?
Yeah. So as a microorganism moves from, I don't know, mouth to stomach to small intestine to large intestine, what other environmental condition changes? Oxygen changes. We've got about 10%, maybe a little bit more oxygen in the stomach of oxygen. Not specific organism, but what do we classify them as? And take a look at your notes. Or the book. They do. That's absolutely true. That's one group. There's another one probably too. Annie? Okay, facultative anaerobes would as well. So both of these have the ability to live in the presence or absence. There's a difference between those two in particular. So let's review these 5 real quick. 5 major types of organisms based on oxygen requirement. Obligate aerobes, meaning they require what? They require oxygen. What does that tell you about their metabolism? It's aerobic, right? They can't, without oxygen, they can't live. In other words, they can't produce energy without oxygen. There are 2 of the 3 major ones that we've talked about: aerobic respiration, anaerobic respiration, and fermentation. There's only one that can occur, or only one that requires oxygen. That's aerobic respiration. So obligate aerobes, like Pseudomonas fluorescens or Pseudomonas aeruginosa, they require oxygen to live. They only perform aerobic respiration. There's obligate anaerobes. They live how? In what types of environments? No oxygen. None whatsoever. What kind of metabolism can they perform? Fermentation. What else? Anaerobic respiration. Now these are broad categories. This isn't going to be true for every obligate anaerobe, But what you're looking for is that an obligate anaerobe may be able to perform fermentation and may be able to perform anaerobic respiration. Definitely, though, they wouldn't be able to perform aerobic respiration. They die in the presence of oxygen. There are microaerophiles. They like small amounts of oxygen, but they still require oxygen to live. So they'll only perform aerobic respiration. And then we've got the last two. Aerotolerant anaerobes and facultative anaerobes. And if you're looking at these tubes here, here's an aerotolerant anaerobe. I'll just call it an AA. And this one right next to it is a facultative anaerobe. Take a look at those two tubes real quick. And what differences do you observe? What's different about the growth pattern between facultative anaerobes and aerotolerant anaerobes? No, that's actually growth. That's bacterial growth. But you're starting to hit on something that's pretty important there. This is my favorite time to bring up. Who has seen the honey badger video? This is like 5 years old. You guys need to watch this.
Cobra?
Huh? Yes. Honey badger versus cobra. The honey badger gives no what? Or honey badger doesn't give a what? Nobody? The honey badger doesn't give a shit. Okay? The aerotolerant anaerobe is the honey badger of bacteria. Why? You guys are killing me.
You said why does it go in there?
No, I said why does the aerotolerant anaerobe is like the honey badger?
Because it doesn't give a shit.
Thank you. About what?
About anything.
About anything. About oxygen.
About oxygen.
All right?
Doesn't need oxygen.
Thank you. It doesn't need oxygen, but what's true about the growth there in tube 5? Oxygen's, so these are thioglycolate tubes, and we'll play with these in a few weeks, or a week or so. Oxygen's at the top. There's no oxygen down here. What's true about the growth of this organism in the tube? It's growing in both, and actually the amount of growth throughout the whole tube is the same. It doesn't care if oxygen's present or absent. Its growth remains the same. It tolerates it, but it doesn't use oxygen as a final electron acceptor. Aerotolerant anaerobes are by definition anaerobes that only perform fermentation and anaerobic respiration. They have additional enzymes, and we'll talk about this either today or we'll cover it first thing on Wednesday. They have additional enzymes that allow them to survive in the presence of oxygen. Facultative anaerobes, on the other hand, are a little bit different. So Emma, what did you note about that facultative anaerobe? I think you started hitting on this. Yeah, but what's true about the growth here versus... Yeah, and another way to describe this, and this might be a little bit different depending on the organism you look at, it's much more robust growth up here than it is down here. Why? Oxygen supports more growth, more ATP production, exactly. So facultative anaerobes, facultative here means they can choose. And the choice, quote unquote, is based on the environment. If oxygen is present, they're going to perform aerobic respiration. If oxygen is absent, they can switch gene expression and now express genes that support fermentation or anaerobic respiration. In other words, our facultative anaerobes can perform any of the 3 types of metabolism that we've talked about. So to go back to gastrointestinal tract for a minute, we're going to find both of those commonly within the gastrointestinal tract. We'll find our foodborne pathogens are oftentimes facultative anaerobes. They are ones that can survive in the normal aerated environment, and as oxygen requirements or as oxygen levels decrease from the stomach to the small intestine to the large intestine, they begin to change their metabolism. They go from aerobic respiration to fermentation or to anaerobic respiration. Those aerotolerant organisms, on the other hand, will always perform anaerobic respiration or fermentation. Okay. Couple of others we haven't talked about yet: pH and salt concentrations. pH ones are pretty easy to define. So, what's an acidophile?
A microbe that thrives in a very acidic environment.
Perfect. A kilophile?
Basic.
Neutrophile? Beautiful. Acidotolerant?
It grows in acidic environments, but not as extreme as acidophilus.
That's true. We might also think of it like so if you're tolerant of an individual, you deal with that individual pretty well, right? Acidotolerant organisms would be ones that grow pretty equally well, anywhere from neutral to about pH of 4 or so. Doesn't matter if that acid is there or not, they grow pretty equally well over that range of pH conditions. Common acidotolerant organisms would be the lactobacillus. They grow well at 7. They also grow well at a pH of 4. At about a pH of 3, they begin to die off. This terminology can be also used for salt concentration. So we have non-halophiles, don't like salt, usually start to be inhibited in their growth by about 2% NaCl. We have halophiles, which like higher percentages, extreme halophiles, which like really high percentages of salt, up to 30–40%. And then we have our halotolerant organisms. And halotolerant organisms will be able to grow at salt concentrations anywhere from 0 to 8, 9, 10% in some cases. I know at least one person picked out a very specific example of one, but there are some that we've worked with in the laboratory. Mannitol salts is a good test to test for actually not only halophiles, but halotolerant organisms. Does anybody remember what actually grew on mannitol salts agar that we used in lab? We had 2 bacteria. We had Staphylococcus epidermidis, and we had the other one was Enterococcus faecalis. One of the 2 grew on mannitol salts. And it was the Staph. Staph aureus, Staph epi, those are considered to be halotolerant organisms. One that can survive can grow over a large range of salt concentrations. All right. I'm going to stop talking because I can see I'm putting everybody to sleep today. So. Here's what I want you to do. Start with this one. Using the terminology that we've been using, I want you to take a look at this one labeled number 2 right here. So an organism grows at room temperature equally well in concentrations of NaCl from 0 to 6%, about half as well at 8%, and does not grow at 10% NaCl. Organism grows maximally at 30. It's rod-shaped. It grows better at pH values from 4 to 5 than at pH of 7. In a static culture, meaning not shaken, it grows at a rate of 1 replication per 2 hours. When shaken, it grows at a replication at a rate of 1 replication per hour. Use terminology to describe this organism. In other words, take the list of terms that we've worked through and apply them to this organism given these experimental results. Right? I'll give you about, we'll work on this one for about 6 or 7 minutes. Feel free to work with somebody else, draw it out if it's going to help. All right, and then we'll go ahead and cap with this one to finish up for today.